My name is Rick Henriksen. I'm a graduate student in Professor Luke Lee's lab. I'm part of a small but growing kind of core of students that are interested in global health diagnostics. And with the microphone right now, Tamina's given me the liberty to kind of take it over and give a little bit of a blurb, a shameless plug for our point-of-care diagnostics idea lab. So we've got a group that founded a couple years ago, and we've got a pretty strong following at Berkeley, and it mainly acts as sort of a lab group meeting for people that are all interested in point-of-care diagnostics. So we bring people from across the campus, and in fact from the Bay Area, because we advertise to a number of outlets. And so we bring people who are interested in the mechanical engineering, the electrical engineering, the public health sides, the medical sides to the problem. About 50% of our talks end up being from students presenting on their specific technologies that they're working on, and they can get really good feedback from a very diverse group. And about 50% are from outside speakers. So a lot of you people here, if you're sitting here, you've kind of got targets on your head for me, because I'm looking for speakers all the time. And as far as the students go, I know that we have a lot of people from Berkeley. We have a lot of people from the small private school down across the bay. And so you guys are all, of course, welcome to come to our meetings as well. And we're going to try in the future to advertise them much more. So that's the end of the shameless plug for our group. And so all the people you see running around with the gold and blue shirts, they're part of the organization. So the Point-of-Care Diagnostic Idea Lab has been pretty involved with this event. So now on to the real meat. So I'm really happy to be able to introduce John McDevitt. I've had the opportunity to meet him a couple of times, once at a Duke conference on global health, and then more recently this summer at a conference in Turkey, which is pretty fun. He got some interesting photos of me there. And so he's had a pretty good background. He's in a really good position right now to teach us a lot about how to develop a point-of-care diagnostic from the beginning towards the end stages. He started with a BS in chemistry, did his PhD in chemistry at that school, and then was a professor at Austin, a co-founder of a product that he's going to talk a lot about, which is the Lab Now product, and currently a professor at Rice. He's had over 150 patents, over 170 publications. He was a presidential young investigator. He's been named by Popular Science as the best of what's new in personal health. And so he's been able to get traction both in academic circles, but also in popular circles as well. Not that we're not popular, but. So his lab is organized around a few different areas. And he'll go into more detail on some of these thrusts, but they've got a saliva diagnostics component to their lab, cardiac risk assessment, HIV monitoring, bioterrorism screening, and toxic metal screening. So they're obviously taking advantage of all of the different areas where you can have an impact using these kinds of technologies. And so as a beginning stage researcher in the field of global health diagnostics, I'm really excited to be here, and I know our group is too, to have the opportunity to be able to interact with all of these people who are kind of the leaders in this field. And so I'm just going to talk a little bit about his system that as it was before, and then he'll talk more about how they're extending the system. So their first product release was geared towards CD4 analysis for looking at HIV immune functioning. They had $34 million in commercial funding, and right now the next step is to try and take this technology and do some widespread distribution. But on the side, they're also developing some companion tests that utilize some of what they've learned from these platforms. And so the CD4 test is also a cell-based test, and they've got another cell-based test for oral cancer. And they've also got some tests that are based on soluble chemistry for, like I mentioned, cardiac disease, some drug abuse, trauma, and other areas. So John McDevitt, I'm going to kind of leave the floor over to you, and you can talk more about how you're taking some of the exciting stuff you've been doing in the past with global health and how you're pairing that up with some new sort of non-traditional global health areas. And I'd like to, again, thank our speaker.
Thank you, Rick, for that kind introduction. Thanks also to Luke for the opportunity to come talk to you here today. Let me move past Piazza San Marco into a discussion of healthcare. And I think as we have all witnessed recently, healthcare has become a topic of debate for our nation. It's like a full contact sport. And this sport has been very difficult to referee, unfortunately. There's only two things that we agree on, and one is that healthcare is too expensive, and we also agree that something needs to be done, but exactly what needs to be done has not been well defined. We need to define the win-win situation. So let's look for a minute at the microelectronics industry for inspiration about how we might best define the win-win. And consider U.S. healthcare costs versus that for microelectronics. And for the past 3 decades, we've seen amazing cost reductions on processor power on the order of 58% a year. There's been no industry like this. It's been phenomenally exciting to watch this evolve. But healthcare is going in the opposite direction, increasing at the rate of 7 to 8% per year. And the per capita healthcare costs have risen from the 1970s, where they were about $350 per person, to now north of $7,500 per person. 17% of our economy. 17% of our economy is going into healthcare now. And this is taking the richest country on the planet and nearly pushing it into bankruptcy. So this is our challenge. And when we look at the disease demographics in the US, in Africa, and also on a global basis, you'll find that each region faces its own set of challenges. We have some commonalities of heart disease in the States as well as heart disease on a global basis. But in Africa, for example, HIV has become the #1 killer there. And in general, there are too many diseases, there are too many gadgets, and there are far too few resources to treat all humans on a global basis. And this slide is a little bit hard for you to see, I think, but it's meant to be an eye chart here also. But basically what I wanted to emphasize here is that in places like the United States, we have in excess of $7,000 devoted to healthcare costs, but 1% of that is available to treat people in Africa. So just wrap your arms around that particular issue, that we need to develop solutions that are much more affordable. Now, let me project past where we are today and just imagine a better future. And this is one where individuals are empowered with non-invasive testing modalities. And these systems exist in the homes, they exist in the hospitals as they do today, but they also move into non-traditional areas, or what we would consider non-traditional today, like pharmacies and also the hut. And the ability to provide the individual with feedback would enable us all to go down a different pathway than we exist today, which is reactive medicine that waits till we have severe symptoms, waits till it's very expensive. And in this kind of situation, we build up new devices like we build up cell phones. We create them with microfabrication techniques. They become dirt cheap. We are able to develop global solutions and amortize the cost on a global basis instead of specializing devices for each little niche of society in each country. So we're not there yet, but if we did go in this direction, we could offer everyone better health care in a more affordable way. So let me now talk about where we are. Project exactly where we're going relative to these ambitious goals. And my talk will be organized as I've shown here. I'll begin with a discussion of global diagnostic solutions and tell you a little bit about our approach, which we describe as the Bio-Nano-Chip approach. And this comes out of a general area that we started 14 years ago based on the concept of a digital sense of taste that can learn. and take multiple different types of chemistries and put that in digital form, create us fingerprints. I'll move into the next step, which I think is critical for this area of microfluidics and lab on a chip, is to begin to move into clinical trials and move past the publication. And I'll talk about briefly 3 case studies and allude to the biomarker highway. We have an information highway now that you're all familiar with, but I'll project a new direction where biomarkers move in a similar trajectory, and then talk at the end about the healthcare cost curve. So first, let's look at this issue of what I describe as the biomarker bottleneck. And today we have 20,000 biomarker cancer papers, and 6,000 of them in cardiac. And so these come out of the genomics, proteomics, glycomics, metabolomics areas, and it's incredibly exciting at one level if you are happy with the publication, but the brutal reality is that the publication is not good enough. And each year the FDA approves 1 biomarker per year. And this is for every disease that affects the US population, 1 biomarker per year. And one of the first lessons I learned in moving to the Texas Medical Center just last year is that if it's not FDA approved, they're not using it. And we're not benefiting from this. So these papers are like this pile of keys until we find the right lock. Now, think about the implications of this. We have information and we can't tap into it. It's as simple as that. And if we look at the information highway, this has changed our lives. And it's to the point now where I think a lot of people in the audience don't know how to use the yellow pages anymore, or haven't gone to the library frequently, because we have instant access to this information. That's incredibly powerful. And the microelectronics and the software industries work together seamlessly. And that's just really, really cool. We have Moore's law that defines a target, and the industry will hit the target next year. And the challenge with the biomarkers is it's based on a series of things that have information gaps, knowledge gaps, technology gaps, and a huge lack of standards. I mean, it boils down to these kind of things. And if we could, though, take these biomarkers and put them into clinical practice quickly, we would change the practice of medicine overnight. And so we need to find a way to do this. And one of the things that we did when I first got to the Texas Medical Center is we set up a matrix of over 100 clinicians, and we analyzed this trajectory, this pathway to the FDA. And there are 3 basic steps: discovery, validation, and clinical implementation, en route to delivery of healthcare to people in the US. But there are a series of roadblocks, and these are dead ends in essence. And these come from a number of different factors. There's an entire talk just in the inefficiencies that we have here. But there are 3 technologies. There are 3 different research groups and or companies involved in this. And there's a problem with poorly executed clinical trials, lack of incentives, lack of consistent intellectual property drivers, and lack of standards for handling samples in biorepositories. These are all roadblocks that get in our way. Now, over the last 14 years, we've been working diligently with a single focus. So we've been like the turtle, moving slowly towards the goal of making affordable and accessible technologies that do one thing, and that one thing is complex fluid analysis. And we began here with an electronic taste chip, and then with time, in essence, what we've done is we've added different types of taste buds into this system, different types of chemistries, and this is represented in these milestones that I mention and feature here, moving to DNA-based tests and to cardiac risk assessment, into white blood cell detection, PCR on a chip, oral cancer, spore detection, et cetera. But don't let me overstep this, because this is a critical step for us in 2004, beginning to reach out into the real world and starting this translational effort. And just last year, in July of 2009, my group moved to the Texas Medical Center, and this is us here at this stage. And this has been a wonderful opportunity now to begin to put these devices into clinical practice. And we've been there just a year, but we're in 6 clinical trials, and we've just recently established the Texas Cancer Diagnostics pipeline with MD Anderson Cancer as our neighbor. So Rice University is a small private university. I come from America's largest public university with a great football team, and Rice needs to choose its battles. And so we don't have a great football team, for example, but we have a very good baseball team. And this is our baseball diamond scientifically now. This is a newly constructed $300 million facility described as a bioscience research collaborative. And it was this building which actually drew us to Houston. And we'd love to have you come visit us. We're on the 3rd floor. Now, I grew up just up the street here in Los Altos, California, and growing up in Silicon Valley, and then moving to Austin, Texas, which is described as Silicon Hills. So the integrated circuit has been with me for a long time. But when I moved into Houston, it really struck me that there was more silicon in our life. And this looks a lot to me like the Silicon Valley of medicine for the following reason. We are within 1/2 mile of the world's largest contiguous medical center. There are 47 different medical programs. There's MD Anderson Cancer Center, that's the world's largest cancer program. There are 93,000 healthcare workers. 2 of the 3 largest cardiac programs are next door. I think if we could get better organized, we'd be dangerous, but at this particular point, we have an incredible collection of expertise clinically, and that's really impacted how we've been able to move our lab-in-a-chip systems to the next level. So this slide, which again is a little bit busy, another eye chart here, but it shows you the type of clinical collaborations that we have going on here. This is really the dream team of clinicians, and we're happy to work with them. Now, a little bit about our Bio-Nano-Chip, and again, coming from a background of the chemistry of microelectronic systems, I think a lot about the drivers in the next step about making healthcare affordable and scalable. And if we go back to 2 key discoveries in microelectronics, 1947, Bell Labs' discovery of the transistor, this particular element did not create a scalable system, but it created the conceptual basis for electronics to take off, the 3-point transistor. Jack Kilby, though, helped at the next level 11 years later by bringing into the field photolithography. So it's the concept and photolithography, something that makes it scale, that is key to this area. And both the concepts and the scalability have to be where we push the microfluidics next. So our grand challenge has been moving across all these different disciplines, from microelectronics and material science to microfluidics to optics to image-based analysis to bioanalytical chemistry, immunology, clinical, and also bioengineering. And in moving to Rice, my appointment now is 50-50 in chemistry and bioengineering. And so I have a legitimate case to spend more time on this, which I didn't before. But I'm very happy to be in a position to integrate, because we need to bring all the pieces together into something that looks like this. So this movie shows our Bio-Nano-Chip in action. And first, you see the card inserted into the analyzer. Barcode reads that chip, and it says what kind, the CD4 chip. And now there's a series of fluid packs here and here, sample goes there, but let me direct your attention to the top. And red and green light-emitting diodes are going off on a strategic time, during which point there is a video chip that's snapping pictures. And that's key because everything that we do is an image-based analysis. And the image-based analysis ultimately goes from what's there and what concentration to move from a macroscopic kind of test into a miniaturized one. So these are all done with a glucose-like interface, and pushing a point that Paul mentioned too, which I agree very forcefully about the need to have non-invasive testing. So we work either with a drop of blood or a few drops of saliva and then do a test that comes out with this kind of report. This is a CD4 count that is more traditionally done on a flow cytometer, but it's coming out of this box. Now, if you dissect a little bit in this interface, it's an analyzer that works with a companion chip that's customizable. And this particular box and the chip can be retasked to different diseases, as I'll show in a minute. But this analyzer is a lot like a laptop computer that you can take anywhere and take new software or new types of chips and then do a new test. And like a laptop, this has a fully embedded PC, so it's a communications station, it's an analyzer, and it's a mechanical, an optics, a software, and a user interface. We've designed the keypad to be like a cell phone, so you text the patient's name into the top of this box, and it's geared towards a 7th grade education so that we can move this outside of the lab and into the backpack and into the hut and into Walgreens and into CVS pharmacy, into your homes, potentially into the future. So a little bit about the Bio-Nano-Chip. I've told you what it has, and it's helpful, I think, to talk about what it doesn't have or what it replaces. And here we've discovered new concepts, microfluidic concepts, and ways that we can capture cells and ways that we can capture soluble chemistries that allow us to get rid of larger instrumentation. So we have high fidelity tests here. We have chips now that measure volumes, and so we no longer need the pipette, we don't need the white coat, we don't need the vein puncture, the vena puncture, stable power replaced by batteries. The cold chain is no longer necessary. We have reagents embedded on the back of these cards with 2-year shelf lives in Africa ambient, and all of these come together to make more sophisticated tests portable. Now, this particular slide gives you some sense about how we gather information, and there's 2 channels, one that comes from the top and a second from the bottom, but they both meet in the middle here at this particular card. On the top, we stain cells. We capture cells on a membrane. On the bottom, we capture soluble chemistries on microsponges that have a nanonet. And so this gives you some sense of the medial slice of this chip, or this particular bead, which is right here. And then if you magnify a little bit further in a space-filling sense, you'll see there's very high surface area. And this provides us with a high-fidelity way to do testing on a miniaturized platform. Now, we want these systems to scale, so a little bit on the materials considerations. We started off with our bead-based sensors using MEMS-based structures in our publications, in our first set of devices. But as you look at this, scaling up to the 1 million copies, the cost per test would never get lower than $10 a shot. And although this might be okay for a genetic test where you pay $1,000 for, it's not okay for a $1 test. And a lot of clinical tests you'd like to move well down here, and plastics and stainless steel can move in that direction. Now, one of the key things that's at the heart of everything that we do is that we look at the big refrigerator and say our device performance is going to be as good as that. And this is a different mentality than point of care as it exists today. Point of care today is more expensive, and it's a compromise relative to the analytical characteristics. It doesn't have to be that way. And the way you get to this point is miniaturization of the system, but not miniaturization of the detector. And so this gives one example here. We have a 200 micron diameter bead that we can put in an array, but it has effectively 10,000 sampling layers relative to an ELISA plate that it might replace. And so we get big signal in this small footprint, and that's a key factor. And this is borne out not necessarily only in the words that I say here, but in the publications that we've had in this particular area. This particular slide shows you a comparison of 13 FDA-approved tests for C-reactive protein, and the approach that I'm describing here lowers the limit of detection by a factor of 3 logs. And that's key now to open up some new kinds of applications in saliva-based diagnostics that I'll allude to in a second. Now, another key theme in the microfluidics area is the world-to-chip interface. This is a big issue. If you have a chip in a lab with lasers and sophisticated instrumentation, you will be on a trajectory to get interesting results, not necessarily to be in a backpack. And that being in a backpack is our goal. But let me give you some sense of our chip in the lab is over here. This is what we published 14 years ago. You see a silicon-based chip for $10. Actually, it cost us more like $1,000 when we made it at the University of Texas at Austin in small quantities. But now we're looking into something that's much less expensive, a stainless steel etched chip, which is both cheaper and actually more efficient. So this is an interesting case where we now tee up the golf ball in a round hole, and we force the fluids through that particular bead in a more efficient way. So we have much less waste, and that leads to a factor of 10 to 100 less reduction in the amount of reagents and the amount of sample volume. And that key step of having a better way to do the world-to-chip interface makes all this possible so that we can do this within a credit card instead of all the instrumentation that you see there. And this just gives you a little sense of that golf ball, golf tee interaction. The round bead on the square pegs has 4 bypasses at the corner. It reduces the pressure buildup, but it also creates a nice drain. And ultimately what we need to do is force the reagents through the bead because we're impatient. We don't want this to take hours. We want this to take minutes. And we can do that with this kind of geometry, which is shown over here. Now, the other thing that I think is key for moving forward is to get to the point where we can model our circuits in a way that microelectronics does. And when you can model the circuits in a fluidic sense, then you're at that position where you can really go past these knowledge gaps. And that's where we are today. We've just developed full models, computational fluid dynamics models, that look at our most difficult tests that are shown here. And your eye can barely see maybe some subtleties here, but I've raised the ceiling here a little bit on this side relative to this side. And now you're looking at the capture of protein for a sandwich assay, which is happening within the bead. And I show this for a couple reasons, because it gives you some perspective of what's happening inside the bead. So this is a 3-dimensional container. It's like a cuvette that's gathering the reagent, but it's also susceptible to how we design the chip, and we now are in a position to model that more carefully. Well, being in California, I thought I'd derive something that maybe you've seen before. This is a spin-off of the nanochromatic ad campaign from Apple, and we've begun to think about developing chips with the same kind of capacity of moving now from disease to disease to disease. And what you see flashing behind you now are the 6 major clinical trials that we're in at this moment. And again, for 20 years in Austin, I wanted to do my first trial, and now we're put in a position to move aggressively into 6. So I'm not going to go into great depth, but just give you some sense of the clinical trajectory for these systems. In the HIV area, we've targeted CD4 detection, and so now the drugs exist in Africa, but we miss the capacity to diagnose the immune function for patients, and without that knowledge, you can't effectively treat them with antiretroviral drugs. So one of the key complications now, and this was published in Nature a little bit more than a year ago, but we've hit the low-lying fruit in Africa. We're delivering care to those individuals that are close to the regional clinic, but between 2/3 and 3/4 of them are not close. And it's not as simple as getting in a sample in a backpack and taking it to a regional clinic. We need to go out to these individuals and deliver the test to them. So the particular test that we need to do is a CD4 HIV immune function, and CD4 is a subset of the white blood cells. And whole blood is a complex mixture of proteins, of electrolytes, of lipids, of a variety of different cell tests. And we need to find the needle in the haystack, and that's shown up here. This one little cell, we've got to get rid of everything else, and we've got to count that in an accurate way. And the way we do this to eliminate the sample preparation that's done here in America is we develop a chip that has a little screening modality. So this is really simple, and we take advantage of a bio capability of red blood cells, which are the majority of the cellular material we got to get rid of, and they work their way through 3-micron holes. The more rigid white blood cells are left behind. An electron micrograph picture of a real membrane is shown here, and you see some red blood cells, you see some platelets over here, but the majority of the cell content left on that membrane is white blood cells. And at this stage, we need to bring in chemistry to bring this to the next level. And so we bring in antibody-based reagents, and we color code the cells. And we do this with respect to 2 different antibodies, CD3 and CD4. When both of those overlap on the same cell, we get a green and a red combination. The camera reads that as a yellow cell. And if it's of the right size, it's about 8 microns in diameter, we count it. And that's how this chip works. And when we do a correlation versus the gold standard, a flow cytometer, we get very, very good agreement. So we basically have a system that can replicate that which exists here in the United States and do that on an affordable system that can go into the hut. Now, a second adaptation now of this CD4 chip is going into the area of oral cancer. This is moving into another clinical trial now involving 3,000 patients. A little bit more than a year ago, we published our first publication here, which describes what I'll talk about being the softball pitch. And the softball pitch, in this case, is having healthy and disease. We needed to begin there, but more recently, we're moving into the fastball pitch. And this fastball pitch is looking at what is really needed clinically, and that is distinguishing between pre-cancer and cancer. And clinically, that's the thing that is so challenging with this particular disease. So this is basically now a reprogramming of the CD4 chip, but it's instead of a flow cytometer that we're replacing, we're augmenting a pathologist. So it's pathology on a chip, but it's another cell-based test. And in this particular movie that you saw go by you, where there's a cocktail of 3 different reagent sets, a red, a green, and a blue, that stain different compartments of the cell. And this serves as the basis for the diagnosis of the oral cancer patient. So you see how our analyzer does this test. There's a series of 3 different image sequence that are stacked one on top of another, and then via software we combine them into a single in-focus image, which is on the top of that image stack. Then what we do is we profile the cell, and we look for the nucleus, we look for the diameter of the cell, and we look also for surface markers. In this case, it's EGFR, which is overexpressed in oral cancer patients. And it is through that combination of 3 different colors in 3 different spatial regions of this cell that allows us to speak a different language than the pathologist, but interpret and lead to the same type of diagnosis. So here is showing some of the morphological features that come out of this. EGFR by itself is not that good. It's decent. It's area underneath the curve. And for those of you that are not familiar with this is basically the diagnostic accuracy. Perfection is 100% sensitive, 100% specific, and it sweeps out an area of 100%. So this is 82% of that. We do better with the nuclear to cytoplasm ratio, but when you get down to it, you want to be as close to perfection as you possibly can, because you don't want someone to go underneath the knife unnecessarily. And we get there at this point by bringing in a combination of the morphology of the cell as well as the surface marker. Now, the last clinical area that I'll talk about is the #1 killer in the US and also on a global basis. And this is a huge, huge expense for our society. If you have a heart attack, you will probably spend 12 to 14 hours in the emergency room at a cost of about $1,000 per hour. There are 5 million people going to the emergency room in the US each year. 80% of them are false alarms. And so this is a great big burden on our society. And if we could rule in or rule out either of those 2 cases more efficiently, it would be a huge savings healthcare-wise. Now, we began this particular effort 8 years ago, and in a saliva-based diagnostics program, we had access to the salivary proteome for the first time. We had a bird's eye view of that. And we thought, oh, that's great. So we're quickly going to develop a saliva-based test for heart attack, and we'll have it in a few years. Wasn't that simple, but this shows the down selection process here, going from around 3,200 proteins to 177, which have better precedent in serum, to then, through PubMed hits, we went down to 32, and then we began to measure those 32, and now we're down to 15, and we're in 15 for a large clinical trial. The reason for making this point is to go back to this issue of the flow of biomarkers, and if you follow this train of thought, we have new information content we're gathering for the first time on this chip, and these are the cases that we're looking at. But if we go back over here and take a peek at what is actually good at diagnosing the patient, it's not the ones that were discovered via mass spec. And in fact, less than 1/3 of them fall into that category. And so what that has forced us to do is rethink at what point we come into the process. And in order for us to create a fast track to the FDA, we're of the opinion that we have to do this with new technologies, to be involved in the discovery, the validation, the clinical implementation, and we do that with the Bio-Nano-Chips that are in all 6 trials that I'm talking about today. Now, the benefit of doing this is that we could be in one clinical trial, being paid to do work for one clinical trial and gather information for the next 2 trials. And in fact, that's happening in the area of cardiovascular disease right now, where our goal is the simple one. It's the heart attack, yes, no, acute myocardial infarction. Is this chest pain patient having a heart attack right now? But if you look at cardiovascular disease, it starts 30 to 40 years before that, and there's 7 different stages of the cardiovascular cascade. Wouldn't it be nice if we could gather information on all 7 stages at the same time? And in fact, that's where we're going in this trial. So we've paired with Christy Ballantine and Bichem Bhaskar. Christy is a risk assessments leader, and Bichem is a congestive heart failure leader. We're in this trial with 588 patients. There are 15 biomarkers, as I mentioned before, 4 time points, 2 bodily fluids, serum and saliva, and a whole host of clinical outcomes, including cardiovascular disease, but also diabetes and a number of comorbid indications. And I think this is key, is that we can gather this information in a well-defined clinical trial and not only go after our first goal, but begin to gather information for our next goal and the next goal. So just to give you a little comfort, because it gave us some comfort in looking at the data from this trial, these are serum-based biomarkers, and serum is the basis through which the heart attack is diagnosed today. So you see myoglobin, cardiac troponin I, and CKMB. These are the dominant biomarkers. They show up early on. In the new bodily fluid, though, what I'm showing you next is virgin territory. This has not been explored yet. And we see now myoglobin, again, being a very nice diagnostic biomarker in saliva. And so we don't have to wait for the vein puncture for this to happen. It can happen in someone's home or in the ambulance. Even more exciting are these 2 biomarkers, BNP and C-reactive protein, which you see have a short time response. And the reason why I'm focusing your attention on that early time response is because this is what we need to do. We need to more quickly diagnose heart attack victims, especially those that are non-STEMI. These are the individuals that are not showing up by EKG, that have to wait about an hour and a half or up to 12 hours for the lab results to come back. And at that point, it's Monday night quarterback. You've either lived or you've died anyways at that stage. And so gathering information early on is key. So I'm getting ready to wrap up my talk here, but let me end with this issue of a little bit on cost perspective. And let me acknowledge it's very difficult to get the fully loaded cost in different environments. But if we make our best projections here, in the CD4 effort, we're looking at an 80% cost reduction for the instrumentation relative to what exists in South Africa today, and a 50% cost reduction on the disposables. Moreover, this analyzer is expandable to other kinds of tests, and it's easy to use, and that lowers the barrier through which the healthcare provider education that we'd have to get involved in this. And these tests are compatible both in resource-poor and more developed countries. So as I close my talk, let me just provide you a brief summary here. We have now developed a number of biomarkers that are based on a number of new sensor themes, and these have a common structure of being in a platform and having elements that allow one to gather more information content as this goes forward. Just like the human being that comes with primitive information about the sense of taste, Sweet is good, bitter is bad. We learn everything else during the lifetime. We are at that stage. I think we're an infant now with respect to where we are in these diagnostic devices, but we're putting them into the clinical trials that I think will allow us in the future to look at new fingerprints. And we've developed these tests both for soluble chemistries as well as cell-based tests. I think we're moving into a trajectory where we can look at a more efficient pathway onto the FDA. This will need to be borne out in our clinical trials, so it's premature to say that with certainty today, but we are seeing some acceleration in the information content that we can get from these trials. We've begun to map out Moore's Law associated with clinical tests. We've just published this in a recent issue of Nanomedicine. And it's not the processor speed, it's not the feature size, but it's the number of tests per person per time that is our information content. And the time element here is key. It's the total time to do the test, to get the test to the lab, for that result to go back to the clinician because they need to act on it and it can't sit in the clinical lab. Again, let me thank Luke and Rick for the invitation, and thank all of you for your kind attention.
So now the floor is open for questions, so you can pull someone with the microphone over.
Could you briefly elucidate the FDA approval process and what goes into this bottleneck for biomarkers?
Well, in 5 minutes.
Then drug qualification, I'm assuming.
Okay, so let me talk just briefly, and maybe we should have a sidebar during a break because this is a lengthy issue. But the lab-on-a-chip area faces new challenges with respect to FDA approval. And it's very tempting to think about these chip-based approaches as being ones that are like the tricorder from Star Trek that can gather lots of information and it comes together as a fingerprint and then you interpret the fingerprint. The FDA has been burned on early releases of multi-marker panels. And so they are now more cautious about releasing multi-marker panels, and that's where we're going here. We want to move past the single analyte silver bullet test. We will, though, do that en route to our steps to the FDA because the FDA has an easier pathway if you take something that they would describe as Me Too or a 510K step, which is a substantially equivalent, so you're basically repeating what's already out there. That particular step, on average, takes about 72 days to get FDA approval to. Bringing in new bodily fluids forces a new kind of test, a new level of uncomfort that the FDA would have. So as we move the cardiac chip into clinical practice through a saliva kind of test, we are going to have to go through a number of additional steps that provide comfort, I suspect, first from a serum-based perspective, and then move on to saliva from single marker onto multi-marker test. And ultimately, one of the things that comes out of this exercise when you look at what the FDA wants, they want you to develop a test where you don't mess around with the mathematical model in the way that we would typically mess around with some kind of model in a publication. But you need to put the stake in the sand at some point and then not move it and look at totally blinded samples. An entire new division of the National Cancer Institute has been formed, the Early Disease Research Network, basically to do this, to make sure that groups that are moving cancer diagnostics are following the right kinds of protocols. And so there are standards, but I think there are also different cultures that are represented in this room. Some are more clinical, some are more diagnostic, some are more lab on a chip, some are more microelectronics, but these are different cultures that I think to be successful all have to learn to take the right kinds of steps that address this FDA approval issue.
Hi, Alan Northrop. It was an interesting analogy with the integrated circuit that you presented. So if you look at it that way, in the 40s, it was basically an analog transistor, and then it became digital through a manufacturing process in the 50s. And the reason we have cell phones in everybody's hand and computers is, I think you'd agree, is because microelectronics are digital and cheap, but mostly because they're digital, right? Obvious. I know the electronics people here think I'm, anyway, no big deal. PCR was invented in 1986. I did PCR on a chip across the street in 1991. So what's the analogy to get a truly digital diagnostics into the hands of people? If you look at the model, we still got 20 some odd years to get there.
Yeah, I think you hit some very good points, Alan. But in order for this to become truly scalable, and so whether it's digital, the digital piece or the three-point transistor ultimately that enables that, I think it's probably both, because you need the transistor to create the infrastructure to run that digital environment. But you need to have things scale, and things will not scale until we put all the sample processing, all the lasers, all the fluid transfer, all the reagents, all the steps into a platform that can go from one application to the next to the next. And so we've taken some steps there, and by eliminating external fluid delivery systems, by putting everything within a lab card, by having something that's adjustable with respect to assay times, and by spending 14 years developing different chemistries, which we've done one at a time, one after another, we are hoping to move into a situation where these things can scale, and they won't scale until they're widely spread and distributed, until the cost point goes down. And so the fat lady hasn't sung, but I think she's warming up.
I just think that some of those processes are just inherently analog. The biological processes are, to get an analog process converted to digital, obviously we did in electronics. I just know how you're going to get those 6 or 7 or 20 or 30 fundamentally biological analog steps into a digital process.
Yeah. Well, I think that this is not a trivial thing to do, but looking into the future, there are a lot of people that are looking at the circuitry of cells and systems biology kinds of applications. And when we can really get to the point where we understand systems biology in a circuit level perspective, then we could look at the protein expression versus disease stage. And after that handoff is given to us, I think we're in a position to take it, because we have the full computation fluid dynamics pieces to go from the protein concentration to the signal on the chip. And so the other part of this too, for right now, we don't have that, and I'm not optimistic that happens quickly. So we are taking a more pedestrian route, but it's still a systematic route that looks at diseased and healthy in blinded clinical trials, and ultimately programs these chips to distinguish in a multivariate sense the healthy patient and the diseased patient. And so it's a process, and I really don't want to trivialize this, because these clinical trials are going to take several years, but I think the key would be to make this process more efficient, to move robotics into the different stages. And that's what we're doing right now is trying to bring the tools that have made microelectronics scalable faster and put that in more central into the biological process.
My name is Octavian Floresco. I'm from Silicon Biodevices. So putting the scaling and cost issues aside, I have a big problem with benchtop analyzers. So I've looked at sort of the history of the distribution of benchtop analyzers, and I can't find one that has had massive market uptake in anything under than 20, 25 years. So I'm thinking of the iStat, I'm thinking of the Colestec, I'm thinking of a variety of different different implementations. And so my big thing here too is that after FDA approval, you're forgetting one last big step, which is distribution, which is decades long for anything that has a bulky desktop analyzer.
Yeah, no, I think those examples are actually really good ones to look at. A lot of times we talk about the glucose case, which has been transformative for diabetic patients, but the iStat, the BioSite.
Do you know of any success stories that have had a ramp that could maybe achieve the kind of investment levels that warrant that market?
Well, let me get back to your first point, because I think part of the problem here with these cases of the point-of-care systems is that they are more expensive and they are compromised relative to the lab-based test. And you can look at this in the BioSite situation, for example. It's in 8% of the emergency rooms, and yet the result is there in 10 minutes. And so why not get that cardiac troponin I information more quickly? And I think if you dissect that from an analytical perspective, you find that the limitation now is all about the limit of detection and the sensitivity of that test. And so with a 1 nanogram limit of detection, that is far inferior to the large Beckman access kinds of instruments that have 0.02. So when you drop off a factor of 50, you make clinicians very worried about letting that particular patient go because it's below the limit of detection for that particular approach. So I think the key here, and this has not been fully vetted, it's not widespread distribution, but if we do as well as the lab-based test, and if we make it cheaper, I think those are the two key themes. And a third element is if we bring in a platform. Instead of having to have 12 different gadgets around your belt, one for blood gases and another for a cardiac test, but you have them all done within the same kind of platform. I think those three elements will be a key issue to market share and moving this forward. But this has to play out, and at this stage, I think we need to take some steps that will be, instead of considered transformative initially, there'll be niche applications, but get some traction that can take us to the next step.
Maybe, I guess, so from looking at the sales models, just the individual sales cycles per selling one benchtop analyzer, they're so long that you need such a large sales force that it becomes very, very difficult. So I don't know, maybe like in my mind, I assume, let's say you assume you have the best sensitivity in the world, you have very low cost, and all you have to do is distribute a $1,000 benchtop analyzer. The sales cycle for that is so long, and the uptake by doctors is so slow that you have difficulty, in my mind. I don't know.
Well, I agree that it's difficult to bring new products onto the market, but I think if we don't take the steps in this community to pull microfluidics into widespread distribution, that we have an untapped resource that would be unfortunate for the cost of healthcare going off into the future. So there's two ways to look at this. One is to look at the big refrigerator and try to make it smaller, And the second approach, which I'm advocating here today, is that we look at the microelectronics industry as another way to build up the devices going forward. And we can respectfully disagree on what will win out. And I think it's important that we look for new opportunities going forward because we need to reduce healthcare costs. It's something that's a big concern for our nation.
So I think at this point we're out of time for questions. I think it's time to move on to the next speaker. But I just want to, again, thank John for coming here. If you have any other questions, you can see him afterwards. But thanks again.